Thank you very much for this introduction. It's so great to be here at the Missouri Botanical Garden. The last time I was here was 25 years ago, and it's just wonderful to see it in such great shape and all this kind of activity on the gardens and the staff. And it's the place feels just very, very alive and exciting. I'm just so glad to be here. It's also really wonderful to be here to see Peter Raven, who was one of my heroes as a student, and to talk with him today and just to interact with him. That's been a kind of a constant throughout my career of just his kind of inspiration and his generosity through the years. It's also great to see Suzanne Renner here, who was my host when I had a fellowship in Germany. It's great seeing Peter Stevens in the back there, who was one of the sponsors of my postdoctoral fellowship. So it's really just great to come here and see a lot of kind of old friends, and also just to meet a lot of the new staff that wasn't here 25 years ago when I was here. So it's just still such a vital place. And also, what was very unique about it 25 years ago were all these connections between the Missouri Botanical Garden and the University of Missouri at St. Louis and Washington University and all the other universities, institutions in the area. And that's still here. That's something which is just so special, that it's just something which is really unique about this place, really among botanical gardens and institutions really around the world. It's really just a special thing that's just great to see that it was here 25 years ago and it's still going on. It's not the same, it's different, but it's still happening. And that's really great to see that. So what I'm going to do today is just to tell you about some of the research that my students and colleagues and I have been doing for almost my whole career at botanical gardens, but particularly over the last 25 years. So this wasn't a project to think, what can we do at botanical gardens? It was just research that we were doing at botanical gardens to better understand plants, and particularly how things that you can learn about climate change at botanical gardens. So it wasn't really starting off as a project about botanical gardens, but after a while we realized that we were using botanical gardens a lot and in different ways, and that maybe that was something that we could share with people. So with my colleagues and I, we've written a number of articles about how botanical gardens can take the lead in climate change research and education. And so that's really what I'm going to share with you today. So just to tell you about some of the research which I'm doing, but the goal isn't just to communicate this research. The goal is to take someone like myself who does not work at who is not employed by a botanical garden, but just to tell you, many of you who work at this botanical garden or maybe other botanical gardens, just about things that you can do. So there's actually incredible resources at botanical gardens that you could do in addition to what you're doing already. So the reason that you want to work at botanical gardens are things that you already know, but I just have to appreciate them, is that botanical gardens are where the plants are. So if you want to do research on plants, botanical gardens are great places because they're all growing there. I mean, if you want to work on oaks or willows or whatever you want to work on, there's lots of species of those genera or families at those places. Like if you want to work on hostas, they have all these hostas growing on the grounds here, or daylilies. If you want to work on daylilies, they have, like I was told, 1,000 kinds of daylilies on the grounds. So if you want to work on daylilies, don't work at, the daylilies in your garden. I mean, work on the daylilies here with its 1,000 kinds of them, and you can do a great project on flowers, leaves, etc. at the botanical garden right here. Also, botanical gardens are where the botanists are. So if you want to learn about plants, I mean, this is where the botanists are. You can collaborate with people, interact with people who are here, and teams of people from places like the Missouri Botanical Garden. If you all work together, you could do great things on plants, on the grounds here, or other places. Specifically also related to climate change. Botanical gardens often have incredible records, and often people sometimes forget the records because a lot of the records are in the library or the archives or buried in someone's office or in the basement somewhere. And at the botanical gardens where I've worked, they often say they don't have many records. But then when you start searching, they in fact have incredible records that mostly people have forgotten about or don't think are important. Some of these records, as I'll describe later, are just actually photograph collections. So people might say they don't have much of a photograph collection, but the photographs they belong to staff members, they belong to people who are friends of the garden, who have their own collections. And those photographs represent incredible records. And then also botanical gardens are places which have lots of visitors. So it's a place where you can educate the visitors about climate change and plants, but also these visitors and friends of the garden can create a huge workforce which can then gather information. So even if the staff are busy, the volunteers can help out and gather this information. So a simple question is, can we see the effects of climate change at botanical gardens, like the Missouri Botanical Garden or other places? Another reason to think about the value of botanical gardens for climate change research is that things are changing at botanical gardens. So botanical gardens aren't the way they used to be, they've changed over time. And so this is just the weather records at the Blue Hills Observatory, which is the closest permanent observatory to the Arnold Arboretum at Harvard, and you can see that the temperatures have gotten warmer over time. And that's true of botanical gardens, and I'm guessing it's true here at the Missouri Botanical Garden, that things have gotten warmer over time here, so that what was happening back in the time of Shaw, many decades ago, is different from now, and therefore you can use the records to look at changes over time. So there are a number of questions that you could use to address with botanical gardens. So one is, how do species and individual plants, even numbered plants, differ in their response to climate change? The second is how the plants at a botanical garden, like this botanical garden, respond to future climate change. Can you predict how they will respond to future climate change? Can you start a study now which will show change in future decades? So among, for example, new staff members, they can think about 40 years here. So how can we protect plants threatened by climate change? And we can do that by looking at the survival and the growth and the reproduction of plants at a botanical garden. And then the fourth question is, how can we communicate the reality of climate change and the effects of climate change on plants to the public? And botanical gardens are ideal places to do that because you have lots of visitors and newsletters and websites and things like that. And one of the things which I've observed as I've been writing review articles and talking to people at botanical gardens about climate change research is that they are really underutilized for climate change research. And the reason they're underutilized really has to do kind of with two things. One is that the staff at botanical gardens, in my experience, they're busy. They already have lots of projects, they have things that they are already assigned to do, and their days are filled up, and to take on one more task of climate change research is just something that people have trouble contemplating. But yet, climate change is this huge environmental challenge, and I would urge people at botanical gardens to try to do more climate change research. And the other reason why botanical gardens are underutilized is that people who are not botanical garden types or who are not trained as classical botanists don't really understand what the resources of botanical gardens are. They just don't realize that you could come to a botanical garden and, again, look at 10 species of elms and 30 species of viburnums in one place. They don't realize the opportunities for comparative research, and therefore they have to be sold on the idea or told about the opportunities of botanical gardens. But they are underutilized at the present time. And actually, I can say that pretty categorically because 2 years ago we did another review of climate change research at botanical gardens, and many people at botanical gardens said that they knew it was important and they were just too busy to actually do climate change projects at their gardens. So there are just incredible resources at botanical gardens. And just as an example of a resource which I mentioned last night in my lecture is not only the records where people have directly observed the phenology at botanical gardens, but also the herbarium specimens which are collected on the grounds of many botanical gardens. And actually, I don't know that. Maybe I should ask Charlotte. So does the Missouri Botanical Garden have a lot of herbarium specimens collected on the grounds of the botanical garden?
Yes.
Okay, yes.
Program document.
Okay, so at the Arnold Arboretum, they have 100,000 herbarium specimens collected on the grounds of the Arnold Arboretum. And so by taking these herbarium specimens, for example, like this herbarium specimen of Vaseyi's azalea. So this herbarium specimen was collected from exactly this same numbered plant. So this herbarium specimen was collected on May 19th, 1938 from this exact same plant. And this plant is in full flower on May 3rd, 2019. So by matching hundreds of these herbarium specimens collected on the grounds of the botanical garden with when the plants are flowering in modern years, you can document the fact that plants flower earlier in warm years than in cold years, and that they are flowering earlier now than they did in the past. So you can demonstrate climate change very effectively with herbarium specimens. I also want to mention that since this is a very public institution, that it's important to have great photographs. And this photograph here sort of looks like a nice photograph, but this is actually a very highly posed photograph. So this is clearly a gorgeous plant with a gorgeous herbarium specimen with nice lighting taken right in front of this plant in full flower. So we actually have about 500 versions of this photograph. But this is just a great photograph. And we tell people about climate change, we have these great photographs which are very carefully posed and arranged that we can give to journalists so that they'll write articles about our story. Or if you're, for example, if you're using in your newsletter or your website, you could have photographs like this. Another great source of climate change information is photographs. And at the Arnold Arboretum, for example, they had about 20,000 photographs in their library, which is a lot of photographs. But it turned out that many of the staff, I would even say maybe not about most of the staff, but many of the staff at the Arnold Arboretum had private collections of dated photographs taken on the grounds of the Arnold Arboretum. And there were many amateur photographers who over decades had taken literally tens of thousands of photographs, maybe even hundreds of thousands of photographs on the grounds of the Arnold Arboretum. And by assembling all these photographs together, we could also tell the story of climate change because these photographs are taken of plants in full flower, not from numbered plants, but at least you can tell the species of the plant. And so these are just photographs taken of fringe trees on the grounds of the Arnold Arboretum. And you can see an old photograph shows the plant flowering on June 20th, and a modern photograph shows the plant flowering on May 7th. And so you can see the plants are flowering early over time, and generally because the temperature is getting warmer. Another great advantage of botanical gardens is that often the same species are grown at botanical gardens all over the world, and there are a lot of botanical gardens. Unfortunately, they're not evenly distributed throughout the world. There's a lot of biases in how the botanical gardens are distributed, but there's a lot of botanical gardens in the world distributed in many different climates. And so it's possible if you're doing observations in one botanical garden, you can create a network of other botanical gardens to make the same observations on the same species, and then you can look at the effects of climate and local variation on plants' characteristics. And so beginning about a dozen years ago, we established a network of botanical gardens to make observations on the same species of woody plants at botanical gardens all around the world in particular climates. I also just want to mention that one of these botanical gardens was the University of Munich Botanical Garden, where when Suzanne Renner was a director, so she was one of the collaborators in this project. So there were 7 botanical gardens around the world, and we monitored the leafing out time of 1,500 species, 1,500 woody species, those trees, shrubs, and woody vines. Prior to the study, as far as I know, the most species that had ever been monitored for leafing out time, so the time in which the plants form their leaves in the spring, was about 70 species. So we went from about 70 species to about 1,500 species, and that's what you could do at botanical gardens because that's where the plants are. So we were able to just get large numbers of species, for example, like this Aeschylus species. So we recorded the day of leafing out as when the leaves had emerged from the bud to the point where you could see their shapes. So what we found is that when you assembled these huge data sets, that they were just in all kinds of patterns which were emerging, which really people had either speculated might happen or there was limited evidence that it happened. But when you assembled this gigantic data set, you could really just see a lot of patterns which related to ecology, evolution, and climate change. So, for example, we found that there were just huge differences in the number of species in terms of when the new leaves first appeared. That's something that we hadn't suspected, that there was more than 2-month variation among species in terms of when leaf out occurred. So the earliest species were leafing out in late March in Boston, and the latest species were leafing out in late May. So much more variation in leaf out times than we had suspected. So for example, and also a lot of phylogenetic patterns. So for example, members of the rose family tend to leaf out very, very early. Oaks leaf out very late. In general, gymnosperms leaf out much later than angiosperms. So that's kind of an example of a pattern. And then a lot of other really striking differences. So for example, deciduous species leaf out earlier than evergreen species, and that's true generally as a pattern, but even if you look at certain genera, like for example, rhododendron, that again, the deciduous species leaf out before the evergreen species. Again, referring to Suzanne Renner, I hope you don't mind my saying this, but Suzanne Renner was able to focus on particularly comparisons between Old World and New World species and find that within the same genera that often the Old World species leafed out before the New World species. So that was another pattern which she and her team were able to pick out. Also, anatomical differences. So species that had diffuse porous stem anatomy tended to leaf out before species that had ring porous stem anatomy. So a lot of interesting patterns like these we were able to discern. We also shifted a couple of years later in terms of focusing on leaf senescence times. And that's really fascinating because with leafing out times, you can very clearly see there's a temperature control of leafing out. You can see all these phylogenetic patterns, ecological patterns, anatomical patterns. And with leaf senescence, almost nothing. So leaf senescence is much more complicated. When plants are undergoing leaf senescence in the autumn, they responded to a lot more different and a lot more environmental cues like temperature, wind, frost, drought, high temperatures, frost events. And so it's a much more complicated picture, and we don't see these same patterns in leaf senescence. And in particular, we don't even see correlations of leaf senescence, the order of leaf senescence between botanical gardens. There's no common patterns in botanical gardens, in contrast to leafing out times, where the order of leafing out is almost exactly the same between different botanical gardens. But leaf senescence times, there's almost no correlation between botanical gardens. Interestingly enough, there's often a correlation of leafing out times within a botanical garden, though. And that's because of a very peculiar difference, a very peculiar situation, which is often closely related species are planted together at botanical gardens, like all the oaks, all the willows, all the walnuts. And as a result of that, closely related species are planted in similar microenvironments at botanical gardens, and therefore they show similar patterns of leaf senescence. So there's this peculiarity that each botanical garden has these patterns, but the patterns are different in different botanical gardens. I also want to mention a very neat new project that has only started about 3 years ago, which is called Phenops. So when we completed this network of botanical gardens looking at woody plants, we realized that there was a big gap in our knowledge. The gap was our knowledge of the phenology of herbaceous perennial plants, that they are really understudied. People know, of course, when perennial wildflowers flower in the spring or the summer, but people really have no idea when they undergo leaf out in the spring, when they undergo leaf senescence in the summer or the fall, and even not that much information about when their fruits are mature. So beginning 3 years ago, we set up this network, which is actually led by IDIV at the University of Leipzig by Christine Romerman. And in this study, we tried to establish a protocol for monitoring the phenology of perennial herbaceous wildflowers, and then to start monitoring them for leafing out times, kind of growth, starting the kind of growth activity in the spring, flowering, fruit maturation time, or fruit maturation time, and leaf senescence time. So this began to be this study at a number of botanical gardens, starting with 5 botanical gardens, and now it's up to about 15 botanical gardens, and looking for patterns of phenology and eventually climate change for all these perennial wildflower species. This project also, besides looking at the phenology of these wildflowers, also has a very strong emphasis on looking at functional traits of these species, like plant height, leaf size, stomatal characteristics, the thickness of the leaves, nitrogen content, etc. And also placing a lot of emphasis on citizen science of getting the public involved in monitoring phenology. And this is just kind of a graph showing some of the results of this phenops monitoring. So it's looking at factors which affect particular characteristics of phenology. So among the important characteristics that we found that came out of this was the variation between gardens, the plant height, the leaf area, but we also monitored a lot of other functional traits which turned out to be not so important. So for example, if we look at when plants begin to flower across these botanical gardens, what we find is that by far the most important factor in determining when plants flower in the spring is how tall they are. So it's something that I really appreciate how important that was. So basically across botanical gardens, short wildflower species tend to flower in the spring, and tall ones tend to flower late in the spring or in the summer. And then also leaf area is also important. It tends to be negatively correlated as shown in red here. And that's suggesting that the bigger the leaves are, let's see if I get this right. The bigger the leaves are, the earlier they are in the spring. So plants that have big leaves tend to flower early in the spring, and plants that have small leaves tend to flower later in the spring. But if we look at how long the flowering season is, it turns out that leaf size is extremely important. So that plants that have big leaves tend to have a short flowering season, and that plants that have really small leaves tend to have a much longer flowering season. So we're relating these functional traits to aspects of plant phenology. What's also very important about this, and it's pretty strange to think about, is that the particular garden, that the variation between gardens is extremely important in determining initial growth in the spring. And also, the characteristic of the gardens is very important in determining the length of the growing season. So when the plants leaf out in the spring and when they undergo leaf senescence in the fall. And this is a strange result, but it kind of makes sense, though botanical gardens really vary a lot in terms of how sunny they are, or whether they're growing the plants in sun or shade, how much they're watering the plants, how much they're mulching the plants. And this has a very big factor on things like when the leaves emerge in the spring and when they undergo senescence in the fall. So a lot of information about phenology is coming out for herbaceous plants from this study. This study is also important because prior to this study, people weren't really sure how to record leafing out in the spring and leaf senescence in the autumn. And one value of the phenop study is developing a very standard protocol for looking at the phenology of herbaceous perennial plants. So there's a lot of things you can do with herbarium specimens and climate change. And, there's just so many things to do. So every time I talk to people, people are coming up with new ideas. We're coming up with new ideas. Just all the things you can do with herbarium specimens. So many of you, I think, have taxonomic backgrounds, systematic backgrounds. And, of course, herbarium specimens are always going to be valuable for identification and classification and for doing floras and monographs. So, keep doing that. That's great stuff, and that's what you should be using herbarium specimens for. But they can also be used for so many things, and we're really appreciating the value of herbarium specimens for climate change research and for other kinds of ecological studies. And just as an example is that you can use herbarium specimens to look at how climate change is affecting stomates and water use efficiency of plants. And I was actually talking to Adam Smith and also to Matthew Austin over the last couple of days, and they tell me that they are actually starting to do the same kind of stuff that our group did about 10 years ago, which I'm really glad to hear because, I think that we want herbarium specimens to be used as much as possible and with different techniques. And so one thing that you can do with herbarium specimens, which I remember back from my time taking plant anatomy many years ago, is that you can take herbarium specimens and paint them with nail polish and peel off the nail polish and look at it under a microscope and look at stomates and look at characteristics of stomates. But it turns out that you can also use this technique to look at the effects of climate change if you have herbarium specimens collected on the grounds of the Arnold Arboretum that have collected at different dates, like from 100 years ago, and then modern herbarium specimens collected recently. You can get these nail polish peels from these specimens and then see if there's any obvious change in the stomates on these herbarium specimens. Also, if you have a very kind director of your herbarium director might let you take little fragments off those herbarium specimens, and you can use these to test for water use efficiency. I keep forgetting exactly how you do that in terms of the equation, but I think it's something like that when plants are under water stress, they do something with water and water or carbon dioxide, and if they're not under water stress, they do something a little differently. If anyone really wants to know, go look at our paper or ask, maybe it's possible that Matt will remember the difference, but you can actually tell the water use efficiency from looking at old plant fragments. Anyway, so you get patterns which look like this. So these are taken from an oak tree. So a particular numbered oak tree from 1931 and 1985. And so you can look at the patterns of stomates. And one thing which we could see, which was pretty evident from oaks and some other genera, is that the number of stomates, the density of stomates has declined over time, but the stomates were getting bigger over time. So they had the same overall kind of ability to take in air into the leaves and to lose water, but we have this changing pattern of stomates over time. And there was no change in the water use efficiency. So there's no evidence that the trees at the Arnold Arboretum are under less water stress over time because they have more carbon dioxide. So this is something neat you can do with herbarium specimens. You can also use herbarium specimen all kinds of ways for looking at flowering time. And this has been done for a while. So we started doing this, for example, in our lab let's see if I get this right here. In 2005, we started using herbarium specimens to look at flowering time and to use these to detect climate change, the fact that plants were flowering earlier now at the Arnold Arboretum than in the past. And you can also use these to look at leafing out time. And this is something that, again, sort of mentioned Suzanne here. So Suzanne Renner, her lab at the University in Munich and our group at Boston University discovered this technique in the same year that you can take herbarium specimens like these, and you can actually see the date of leafing out. So you can see that in a specimen like this, the leaves are small, they're thin, almost translucent, they're very pale in color. So these are young leaves. So not only you can tell the flowering time of trees and other plants from herbarium specimens, but you can actually get the leafing out time, which is something that people, until we had published our papers, people had not really thought that this was possible. So what we found in our research in Concord, Massachusetts, which I spoke about yesterday, so in Concord we're seeing that plants are flowering earlier over time, that they're leafing out earlier over time. But one thing, after working in this system for 20 years, we were able to determine that the sensitivity line, that the slope of the line of trees leafing out in the spring is steeper than the line of how plants, wildflowers, are responding in terms of flowering. So these lines are slightly different, but they're statistically different. This means that the window of full sunlight that wildflowers have in the spring when they first leaf out, that window of full sunlight is actually narrower in warm years than in cold years, and that it's also getting smaller over time and will be smaller in the future. So that window of full sunlight that wildflowers need to really fulfill their energy budget is getting narrow over time, and it's getting smaller over time. And we were very curious to determine if this pattern, which we could detect in Concord, Massachusetts, using the records of Henry David Thoreau, if that was really kind of an artifact of Concord or only happening in Massachusetts, or whether that was really a widespread pattern. And so what we did is we decided to see if this pattern was really occurring in eastern United States, across eastern United States, and really even in other temperate areas of the world. And so what we were able to do in this modern era, when millions of herbarium specimens are being digitized and made available online, is that we took, for example, in eastern North America, we've got thousands of herbarium specimens of trees with young leaves and wildflowers with flowers on them, who are 6 common tree species and 6 common wildflower species. And we saw when plants were leafing out, when the wildflowers were flowering, and we were able to determine the temperature of that place in the year it was collected. So we've got all this temperature information for all these herbarium specimens of trees leafing out and wildflowers flowering. And then we did this, had colleagues in Europe who did the same thing for Europe with 6 common species of trees and 6 common species of wildflowers. And again, we had colleagues in China who got this same kind of information from China, again, all using digitized herbarium specimens, so people could just do this from their offices without going to the herbaria. And so we got, again, huge numbers of specimens and similar kinds of data across 3 continents to see if this pattern that we detected in Concord, Massachusetts was really generalizable. And these are the kinds of results that we got. And what you can see here is that, first of all, if we look at the temperature in the spring for all these sort of figures here, and the scales are slightly different here. So we have different scales here going from -5 to 20, -5 to 10 here. So if we look at just eastern Massachusetts, or eastern North America here, eastern North America, you can see that when it's a very cold spring, the plants flower and the trees leaf out late. And when it's a very warm spring, the trees leaf out very early and the wildflowers flower very early. But what you can also see is that these 2 lines, that the tree slope is slightly steeper than the wildflower slope, and that window of full sunlight is getting narrower over time and is predicted to get narrower in the future. So this observation that we got from Concord, Massachusetts is really true all across eastern North America. And if we look at Europe and if we look at Asia, what we see is that pattern is not true in those different continents. So that pattern of trees responding more strongly to climate change, it's true in eastern North America, but it's not true in Europe and it's not true in Asia. And so this suggests that something is happening different in terms of the biology of species on these different continents. And this is something that people have speculated about generally, but again, kind of Suzanne Renner and her student Konstantin Zahner have written specifically about how the different climates in these different continents might be affecting phenology differently. What's also very interesting about these figures here is that if you look at the R-square values, they're really quite different in these different continents. That in eastern North America, that the species phenology, spring phenology, is very strongly correlated to temperature. When it's warm, things happen early. When it's cold, things happen late, with a very high R-square value. Whereas for East Asia and for Europe, we again have these patterns, but the correlations are much weaker. Statistically significant, but much weaker. That these species are not just responding to temperature, they're probably responding to other things also, most particularly photoperiod. And this again shows how we can take the data from eastern North America and look at it in more detail. So if we take eastern North America and we divide it up into kind of cold, medium, and warm regions, and look separately at the slopes of the relationship between phenology and temperature, what we can see is that if we just look at the trees, for example, that in the southern United States, the species are responding much more strongly to temperature than in the northern United States. And if we look at the wildflowers here, they basically have a lot of overlap between the wildflowers in the kind of the cold, the medium, and the warm areas. And so really it's the main difference between trees and wildflowers we can actually identify to really the warmer regions, the southeastern part of the United States, not really across the whole eastern United States. So I've mostly been talking about botanical gardens, the things you can do with botanical gardens and plants, but another tremendous resource that's available to botanists is also long-term weather records from phenology gardens. So not botanical gardens, but phenology gardens that are in particular places. And that in many areas of the world, but especially in Japan and Korea, there are extensive phenology gardens associated with weather stations. And this picture here of South Korea just shows where there are phenology gardens in Korea associated with weather stations, and the ones shown in black here are 80 or more years old. And the oldest ones from places like Seoul are actually, this is their 100th year anniversary this year. So there are phenology gardens which have been recording plant phenology for more than 100 years at these locations. And this represents a tremendous opportunity to investigate climate change, the effects of climate change on plant flowering times. And what do we get at these botanical gardens, in Korea especially, is that we can see, again, the effects of temperature on phenology. So if we look at the date of first flowering in relationship to spring temperature, what we can see, for example, for the Korean forsythia here, is that when it's very, very warm, the plants flower early, and when it's very cold, they flower late. But what's really neat about this figure here is that that figure for forsythia is for a 100-year period. The line there, the red line, is the line made from the first 50 years of data. The dots there are the dots from the second 50 years. So what's really neat about this figure here is that it's showing that the predictions made from the first 50 years of data exactly predict how the plants will respond in the second 50 years of data. So that's really neat. Another neat thing about it is that it's also showing that these responses are totally linear. There's no evidence of any kind of curvilinear response or that species reaching some kind of asymptote. They're responding linearly to temperature, and that linear models are more than sufficient for analyzing these kinds of plant responses. I also want to mention that citizen science is extremely important for botanical gardens for these two reasons I mentioned. One is that it represents an opportunity to increase the amount of people making these observations, because we know the staff is very busy at botanical gardens, so it's a way of just increasing the amount of people, the amount of eyes looking at the plants and recording this information. But it's also a way of educating the public and really engaging them in the topic of climate change. And the Royal Botanical Garden in Edinburgh is a particularly successful example of this, where they just had a very huge program of citizen scientists making observations on their rhododendron and azalea collection. But I've also been working with a colleague in Denmark to just see how we can combine different kinds of data, including citizen science data, to really get a much more extensive picture of the effects of climate change on plants. So as botanists, we are increasingly comfortable about using herbarium specimens. So if you look across Denmark and where herbarium specimens are collected, and then what factors are affecting them. So this is actually a study done with allium, sambucus, and aesculus, so onions, elderberries, and horse chestnuts, or I guess you could also say buckeyes. So since we're in the Midwest here, I guess. So if we look at how temperature is affecting these species, I mean, it's a pretty clear kind of temperature response curve that we've seen in lots of places. And again, this is where the herbarium specimens are collected of these three different species. And so we can think about what other kinds of data we could potentially combine with them. And so in Denmark, there's increasing use of iNaturalists. And so, no, excuse me, not iNaturalists. There's increasing use of a citizen science network to make observations. But this citizen science network, people were told to go out and observe the first flower. So they were told to go out and see when the first flower was open. And so this is a record of where people were going out and sort of making these first observations in the citizen science network of where plants were first flowering in different places in Denmark. And this is what the data looks like. And you can see there's a lot of data points out there, almost as many as the herbarium specimens, but they're all kind of all over the place. And that's because the first flowering time is a very weird thing. I mean, people can go searching for them in a driveway, or they can be kind of looking for what they think is the first flowering in some cold place. So we don't see the kind of strong temperature effect that we see. And it's also affected by sample size. In contrast, when we look at herbarium specimens, these are collected mostly when the plants are in full flower, rather than the first events. And if we look at iNaturalist data, so another thing which has come into Denmark in the last few years in the same way in the United States is people going out taking photographs with their iPhones and then trying to, just see what it is. But what they're also doing at the same time they take this photograph is that they are contributing that photograph to this online database. And what my colleague Natalie was able to do was to just realize that you could take these iNaturalist values and also look at how they were affected by temperature. And so what you can see, even though the range of temperatures is relatively narrow because it was only taken in 1 year, so in contrast to the herbarium specimens which were collected in cold years and warm years, so these iNaturalist observations were only made in the range of 1 year, we again see this kind of slope. So with each one of the particular species, it shows this slope of earlier flowering in warm years. But what you can do, interestingly, is you can see that you can combine these two kinds of data sets. You can take iNaturalist observations, which are mostly taken of plants in full flower, and you can combine them with the herbarium records, which are collected when the plants are in full flower, and that gives you just a much higher density of observations, which gives you much greater ability to detect the effects of climate change. So that's one thing which is really neat that people in botanical gardens can do, is you can take your historical observations of herbarium specimens or old records, and then combine them with this more recent kind of material of iNaturalist photographs. Or potentially, you can get people to wander around your botanical garden and just take lots of photographs with their smartphones, and then use the iNaturalist data from this particular location to combine with herbarium specimens. So another technique which is just really underutilized, but is just so effective for determining the effects of climate change, is what we call the dormant twig technique. So what we have done in our lab is that you can go out in the winter or very early spring, but ideally the late winter. You go out in the late winter and you can collect dormant twigs of plants and put them in a wide range of conditions. So you can put them under different lighting regimes, you can put them in different temperature regimes, and this can tell you to what extent plants have overcome their winter chilling requirement at a particular point. It can tell you how much warmth they need to respond in the spring, and it can also tell you whether there's a photoperiod requirement. This is an incredibly simple technique, really easy to do, needs minimal equipment other than having some growth chambers with different temperature or lighting controls. And you can use these to see how plants are able to respond to a changing climate. And botanical gardens, like this botanical garden, is an ideal place to do that because you can have large numbers of closely related species all growing in one place, and you can collect the twigs of all these related species and see to what extent they're responding in a similar or different way to all these different environmental cues. What's also great about this kind of technique is it's not only scientifically very interesting, but it's something that kids can do in a college laboratory if they want to investigate climate change, or kids can do in high school or homeschool projects or science fair projects. It's a real easy thing, for example, to collect a lot of twigs and then just in your house put them in the cold place, in the warm place next to the furnace, out in the garage, and see how these different temperature conditions affect when the twigs will break dormancy and start leafing out. And just this kind of an example of this is that we collected crab apple twigs in Boston it's kind of a little bit hard to read here, but we collected these on March 13th, 2022, and we just put them in the range of temperature conditions which we had in our biology department growth chambers, all these different chambers in our building. So these were all the different temperatures which we had in our building. We just put jars of water with twigs in them in these different temperature conditions and just saw how the plants would respond to them. So this is 4 degrees centigrade, very cold, 25 degrees centigrade, very warm, and this is how the twigs looked after 2 weeks. And so you can see that when the plants are in really cold conditions, they sort of remain mostly dormant or started to leaf out a little bit. When it was really hot conditions, they were already sort of past flowering at that point. It was almost a little bit too hot for them. But in these kind of intermediate conditions, the plants were sort of leafing out and flowering. So there's this whole progression of development responding to temperature. And this is just a very clear way of demonstrating that plants respond to temperature in the spring. A very clear experimental way, but again can be used to contrast different species and can be used for all kinds of different purposes. So where are we in terms of thinking about the future of botanical gardens and botanists in terms of researching climate change? Well, the one incredible opportunity that I mentioned, sort of just one example of, but it's just an unlimited opportunity, is the use of digitized herbarium specimens. This is just a golden moment that we're in right now, where botanical gardens around the world, including the Missouri Botanical Garden, are just digitizing herbarium specimens flat out. And you can do incredible things with these specimens, and we're in this golden moment right now where there's so many new and exciting things to do with these digitized herbarium specimens. So exciting: ecological problems, evolutionary problems, ecological problems, climate change problems, and you can do them now, and it's an exciting time. You can do very original research with these newly digitized herbarium specimens. There's a lot of really exciting things you can do with climate change research because the climate is changing. So, for example, the conditions are getting hotter and drier here in Missouri and other places. And so it's an incredible opportunity, a time in which it's important to think about the effects of drought and heat waves on plants. So you could be going out on the grounds and looking at how heat waves during the summer affect plants, particularly maybe plants from more northern climates, plants that are more used to growing in wet places, and how they're reacting to their environment drying out and getting very hot. Or, for example, we have a situation of warm temperatures in the spring and then frost events. So how do plants react to these late frost events? So what is the degree of frost tolerance of plants? Another interesting opportunity right now is use of functional traits. Increasingly, functional traits are being put in online databases, so it's possible to look at how functional traits are affecting different kinds of phenological characteristics of plants on the grounds of the Arboretum. And then the big unknown in a lot of plant phenology is leaf senescence in the autumn. This is a field which is just so poorly understood. We're really just starting to get good papers on this subject, but there's so much that isn't known about leaf senescence, about how plants really, what cues plants are using to undergo leaf senescence in the autumn, and how that affects their overall energy budget. Another important opportunity for botanists is to link up with ecosystems ecologists. So one of the consequences of climate change is that we're getting a better understanding of the role of plants of absorbing atmospheric carbon dioxide and withdrawing carbon dioxide from the atmosphere. This is rejuvenating ecosystems ecology. And there's a great opportunity for botanical gardens and botanists to really interact with ecosystems ecologists to understand how different plants are withdrawing carbon dioxide from the atmosphere and to what extent they can contribute to this particular important part of climate change. I put this particular photograph here because this is a really strange photograph. This is a photograph taken in August in Boston during a year of drought. And during this year, it was very hot and extremely dry. And these are all birch trees here, black birch trees, which are undergoing leaf senescence and dropping their leaves in the middle of August. So it looks like October, but it's actually August. So what does this all mean? Well, I hope I've convinced you at this point that there's a lot of exciting things to do at Botanical Garden. Probably I've been preaching to the converted. A lot of you probably already know it. But I think that I find Botanical Gardens to be really exciting. I think there's a lot that can be done on climate change research at Botanical Gardens, using all the records at Botanical Gardens, the diversity of species, the knowledge that all of you have, the opportunity of working with citizen science, especially the opportunity of working with herbarium specimens and with these kinds of phenology records from related institutions. I also want to put a particular pitch here that more work needs to be done on living collections, that at many botanical gardens, they have these incredible collections, and all the botanists are kind of working on herbarium specimens, and they're not so much out on the grounds working with the living plants. And so I would all urge you to think about things that you would actually do on the living grounds with the collections here at the Missouri Botanical Garden or other botanical gardens. And also, another interesting opportunity that I probably could have given a whole lecture about is the value of resurveying historical floras. So if you survey the people at botanical gardens have the knowledge to go out and resurvey areas that had floras done in the past. So this is another opportunity. And of course, the importance of involving the public. I have a picture of Casper Bauhin here because recently I had the opportunity of visiting the University of Basel, and I learned more about Casper Bauhin. Of course, I heard about the genus Bauhinia, but of course, I'm pretty sure it's named for him. And but Caspar Bauhin, who lived in the 16th century, I mean, he just did all these great things that we could be revisiting right now. So he did a lot of great things that we could be more like Bauhin and do the same things that he did, but also he did a lot of things that we could come back to again right now. So he established a botanical garden and kept a lot of records from the botanical garden. So, you know, botanical gardens have this historical quality that can be revisited to see how they've changed over time. Bauhin also did a flora of the Basel region, so one could redo the flora of this botanical garden, of this region, to see how the flora has changed over time. And also he was very actively collecting herbarium specimens, which could be restudied to see how the collection date and the characteristics of the plant had changed in these intervening 500 years. So again, kind of looking at a historical figure like Bauhin can give us a lot of ideas about how to be better botanists and to do better research. And with that, I think I'll end my talk, and thanks very much for your attention.